Hello everyone, welcome to Toppr.com. So we are back again with another live session. I am Pushpendu, biology subject matter expert at Toppr.com. So we have conducted another live session in a previous week, right? We had talked about the chapter photosynthesis in higher plants, and that was our first part of that chapter. So today we are going to continue till the point that we have learned in our last class, correct? So before starting with our session today, let's talk about let's quickly revise what we learned in our previous session. Okay, so in this chapter photosynthesis in higher plants, we are concerned with the topic photosynthesis. Okay, so we have talked about this terminology photosynthesis. Photo, the word came from photon, which is from light. Okay, so and synthesis means making something. So photosynthesis is the process in which something is being made with the help of light, correct? So this is basically ये वो process है जहाँ पे plants make their own food. Okay, we have talked about this. Next, what did we talk? We had discussed about some of the important experiments. Okay, we have talked about some of the important experiments, and what were they? We So we had talked about some of the experiments, okay? And why were those experiments we had discussed? We had discussed these experiments so that we understand what was the basis by which scientists thought about this process of photosynthesis, correct? We tried to explain why the basic raw materials or what are the components which are required for the process of photosynthesis. What we studied? Carbon dioxide, so we had talked about the requirements for the process of photosynthesis, correct? So in that process we have seen that carbon dioxide is necessary. In addition to that, water is necessary. And what is necessary? Sunlight, correct? Sunlight was necessary. And also we had talked about a very important component of the plants, that is chlorophyll, correct? So these were the requirements for the process of photosynthesis. And what is photosynthesis? That is the process in which food was being prepared by the plants. So as we continue with the discussions, so you can keep posting your doubts. I'll try to solve your doubts, correct? Also invite your friends so that they can also get helped by our sessions. So keep posting your doubts. We'll discuss on those doubts. So let's continue. So we, through the experiments, tried to prove that these are the components which are required for the process of photosynthesis, correct? So various experiments ke through we had seen that these are the components which were required for the process of photosynthesis. So this process of photosynthesis we had discussed, we had seen the structure, okay? If you remember the structure of chlorophyll, correct? We had drawn this structure. So chlorophyll, it is a double membrane bound structure, We had seen certain structures like this disc shape. These are the thylakoids, stacked thylakoids, they are called as grana. This part is called the stroma, correct? And these thylakoids are interlinked by the stroma lamellae, correct? So we should remember these parts. Now let us try to discuss what are the important things happening in this chloroplast. Okay, so we had seen that this chloroplast is essential for the process of photosynthesis. So we are going to discuss about these processes, correct? So let's see, we had already discussed about some of the pigments, correct? Pigments related to photosynthesis. So what were the pigments we had talked about? We had talked about chlorophyll. We had talked about chlorophyll A, chlorophyll B, chlorophyll C. So these were the three types of the pigments that we had discussed. Now we had talked about that which is the most important chlorophyll which is involved in the process of photosynthesis. We had seen chlorophyll A, this is the most important chlorophyll, correct, if you remember. Now also what had we discussed? We had discussed that during this process of photosynthesis, what happens that these chlorophyll, they act as the reaction center, correct? Reaction center. Now we had talked about two types of reactions. If you remember, we had talked about light reaction and we had talked about dark reaction. Okay, light and dark reaction. So what were these reactions? We had seen, हम लोगों ने discuss किया था कि light reaction was that reaction in which light is required. This component is very much required for the process of that reaction to perform the reaction of this process, correct? So light reaction involves all those chemical processes in which the light is required. So here the dark reaction is a bit of misnomer. We had talked about it. Why it is a misnomer? Why it is an anomaly? Because this term can misguide us. Because dark reactions मतलब वो reactions नहीं जो dark में होता है. Yeah, exactly. The thing is, ये जो reactions है जो photosynthesis में dark reactions which we say are those reactions which do not require light for the chemical reactions, correct? Let me highlight once more. So these are the reactions which do not require light. So in this reaction, what happens? The products which are being made in the light reaction, those products are being used as the raw materials in this dark reaction, correct? You got it? So जो भी components इस light reaction में तैयार हो रहा है वो act as raw materials for the process of this dark reaction, correct? So here we also talked about two important systems. So from now we are continuing with our class today. So this is, we have talked about the photosystem, and it can be abbreviated as PS. Now photosystems, we had seen there are two types of photosystems that are available. Photosystem 1 and photosystem 2. So these are the two different photosystems which are available. Now we'll talk about an interesting fact. Let's start our discussion with the light reaction first. So we'll talk about the light reaction. Okay, so yes, we'll talk about the light reaction. So let's start a discussion. Light reaction. Right. So in this process of light reaction, we will talk about these two systems, photosystem 1 and photosystem 2. So these are actually called as light harvesting complex. Why? The question is why? Why these are called light harvesting complexes? So these are the portions, these are the areas in which light, we had seen that photosynthesis requires light, sunlight. Now light has to be harvested. Otherwise, where will that light be used? Today, we'll see where light is actually used. So light through this LHC, it is abbreviated as LHC, light harvesting complexes, what happens? These complex, they can absorb the light wavelengths. Okay, so I repeat, ये जो LHC है वो light के जो wavelengths है, light energy वो absorb कर सकते हैं. Correct? Now यहाँ पे जो photosystem 1 और photosystem 2 है, इसको reaction centers कहते हैं. Why? Let's try to understand. एक reaction center में, okay, in one reaction center there is chlorophyll A. I'm abbreviating as CHL. Okay, you can also use this abbreviation, but it's better to write the full form. Chlorophyll A, this is, we are saying, the boss. Why? Because this is the main component of each and every reaction center. Correct? This is basically the antenna molecule. We can talk about it as the antenna molecule. Now, we had talked about other pigments also, correct? Like xanthophylls, carotenoids. So what are their functions? Let's try to draw. So if you can see properly, chlorophyll A, correct? If this is the chlorophyll A, this is the boss. Now, surrounding these are the other pigments like xanthophylls, carotenoids, and other various other pigments. They are called the accessory pigments. Accessory pigments. Now, why accessory pigments? Because these are directly not involved in the photosystem. Okay, in this photosystem they are not involved directly, but what it does, it captures sunlight, light energy, and it actually I've got a question, let's see. Okay, so are we discussing why chlorophyll A is most important? Yes, let's see. We are actually discussing why chlorophyll A is most important. Now, so chlorophyll A, we'll see actually why chlorophyll A is most important. But before this, why accessory pigments are important? Because accessory pigments helps in harvesting the light energy. You know, this chlorophyll A alone cannot absorb the energy, and the amount of energy that will be absorbed by chlorophyll A would not be sufficient enough. So what it is done is that these pigments, they also help in absorption of the energy, and that is that light energy harvesting complex. Okay, they harvest the light energy and that is passed on to chlorophyll A. You understand? So they also help in absorbing the energy. Now, also, in addition to this, what happens? These accessory pigments actually help in preventing the photo-oxidation of chlorophyll A. Now, I'll explain what is this photo-oxidation. A photo-oxidation, this is a process in which this chlorophyll A, because of the presence of excessive amount of sunlight, it gets oxidized and it loses its power or loses its function. So in order to prevent excessive amount of sunlight, specifically in areas which receives direct sunlight, excessive amount of sunlight should be avoided. So इसीलिए accessory pigments are very important. Now as you asked that chlorophyll A, now we'll talk about chlorophyll A. Chlorophyll A is actually the reaction center in which it absorbs the energy, and we'll see a system called electron transport. Electron transport. Very interestingly, this is the reason why the energy is produced. Confused, right? So we'll see why and how this process is happening. So here, this photosystem, these two photosystems, photosystem 1 and photosystem 2, they are slightly different in their characteristics. Why? First of all, the basic characteristics, you have to remember, photosystem 1 and photosystem 2. Photosystem 2, it absorbs 680 nanometers of light. You understand? 680 nanometer. So this is the wavelength. If you are aware of the term wavelength, so it can absorb wavelength of light which is 680 nanometers in range. If we talk about photosystem 1, Photosystem 1, it absorbs 700 nanometers. So this thing we have to understand properly. So here is a basic difference in which photosystem 2 वो absorb करते हैं 680 nanometers and photosystem 1 वो absorb करते हैं 700 nanometers. So remember this difference. Now how can you remember? I can help you. Like photosystem 1, it is the second one. Remember, the second one absorbs the lower wavelength. The first one absorbs the higher wavelength. Okay. Now we can continue with our discussion. Now we are going to talk about an important process, which is the electron transport. Let's see what is that. So we'll talk about the electron transport. So let's talk about electron transport. Okay. So in this process of electron transport, what happens? These photosystems are involved. Now, the PS1, photosystem 1 and photosystem 2 we have talked about. These two photosystems are present in the grana. So we have talked, these are stacks of thylakoids, and in the membrane of these thylakoids, these two photosystems are present, photosystem 1 and photosystem 2. Correct? Remember this. Now, let's talk about the electron transport. What happens is, we can draw it. So first, this is photosystem 2. Photosystem 2, what will be the wavelength? It is 680 nanometers, correct? And photosystem 1, if you can see properly, photosystem 1, what should be the wavelength over here? We have talked about, it is 700 nanometer. Now, sunlight, so this photosystem I'm writing, so this is actually the chlorophyll molecule and the reaction center that we have talked about. So sunlight, you can represent it as this, H mu. You can say it, in physics we represent sunlight or light energy as H mu. So sunlight, when it falls, and if the range is 680 nanometers, then it results in the release of what? It results in the release of electron. Let me repeat. So when sunlight falls in this reaction center of photosystem 2, then because of the high energy of this wavelength, the electrons present in the outer shell of these molecules, they are excited, they gain energy and they are released. You understand? Because of the energy, electrons of the outermost shell, they are released. Now, very importantly, these get excited, and here we have electron acceptor. Okay, we have the electron acceptor. Now this high energy electron, which has been released because of excitation, is now accepted by this electron acceptor. We'll talk in detail what are the electron acceptors, correct? Now this electron acceptor now passes on to another electron acceptor. Okay, let us represent it as X maybe. So slowly and steadily down the gradient, or you can say with the system in which the reduction potential, or you can also say it as that the capability or capacity to accept electrons, कितना ज़्यादा इलेक्ट्रॉन के लिए affinity है, उसके basis पे these electron acceptors are arranged. So let's repeat once more. So because of this high energy, electrons are released. It is accepted by the electron acceptor. After accepting, this electron acceptor will pass on the electrons to next one, which has higher affinity for the electrons, correct? So in this way, it is being passed on to what? Here, to photosystem 1. Now this electron is again, when it receives sunlight of 700 nanometers, it is again excited and it is again received by another set of electron acceptor. Got it? Now again, it slowly, based on the acceptance capability with other electron acceptors, it moves on and it results in the production of what? It results in the production of NADPH. From where? NADP plus H plus. Okay, if you can see it over here. So it will be NADP plus H plus. Now, so So this is basically formed, NADPH is formed from NADP plus H plus, correct? So it forms NADPH, correct? So this is the overall reaction. Now very interestingly, you see how it happened. It happened in such a way, so I hope you can see it now properly, correct? Yes, we can see. So here it happened in such a way, you can see, can you see a Z being formed over here? Is it a Z? See, correct? So that's the reason it is also known as Z scheme. Remember this. So this is a very important part which is being asked in your examination. So what is Z scheme? So Z scheme is basically the process in which electrons are transported or transferred through both the photosystems, correct? Now you have to remember that the photosystem 2 starts first and then photosystem 1. So the electrons released from the photosystem 2 is being accepted, is being passed on photosystem 1, and in this process NADPH and H plus is produced. NADPH is produced, which is basically required for the production of energy. Okay, so now the basic question is, from where this electron is coming? From where will the electron come? We'll see. In this process, what happens? Water molecule, it is being split. So I'll draw it. In this process, two water molecule is being split to 4H plus O2 plus 4 what? Electrons. Got it? So this is called what? This is called photolysis. Now, actually, because of this sunlight and certain factors which you need not know the name of the factor right now, because of those factors and the energy from the photon, this water is split into hydrogen ions, oxygen and electrons. So these are the electrons which, because of the splitting, they get excited and it is being passed on in this Z scheme. Correct? You got it? So now let's see in detail what are the components which are present in this process of photosystems. We'll discuss, as I've told you, we'll discuss in detail. So let's see. So let's first talk about photosystem 2, PS2. Here we have seen 680 nanometers. Correct? It gets excited and this is passed on to whom? Here the electron is passed on to a molecule called, it's abbreviated as PHE, and this is basically phaophytin. Okay, you should remember phaophytin. It may not be asked in your examination, okay, like in your CBSE examination or board examination, but it can be asked in your competitive examinations. So it's basically this phaophytin. So phaophytin, this is actually required. Now this phaophytin has accepted the electron. Perfect. Now this phaophytin will then pass this to a next molecule which is abbreviated as PQ. This is plastoquinone. You remember, this is plastoquinone. Now, very interestingly, this plastoquinone in this process, it forms an ATP molecule from ADP plus PI. Organic phosphate and ADP, it is being combined by the plastoquinone with the use of electrons. You should understand this electron is being now passed. With the help of this electron, okay, what happens? This ATP is produced. Now simultaneously it is being passed on to a complex which is cytochrome B and cytochrome F. This is a complex. Okay, this complex accepts this electron from plastoquinone and this will now transfer to PC, which is called plastocyanin. You got it? So from this cytochrome B and cytochrome F, it is being transferred to plastocyanin. So now let's repeat once more. Because of this photolysis of water, water molecule, photolysis, it has released electrons. We have seen 4 electrons are released. Now these electrons are being accepted by the electron acceptor, which is phaophytin. Now from phaophytin, it is being transferred to plastoquinone, which is abbreviated as PQ. In this process, ATP molecule is formed. You have to remember this. Next, it is accepted by this complex, cytochrome B, cytochrome F, and then it is passed on to the plastocyanin. Now, from plastocyanin, this is being passed on to photosystem 1, which is of 700 nanometers. Now it is being passed here. Now from here, when sunlight comes, again it is being excited. So we can use a different color. So it's excited here, and it is being transferred to a complex which is mostly a type of chlorophyll, okay, which has acceptance of P700, that is light of 700 wavelength. So this is also an electron acceptor, okay. So from here, it is being transferred to quinones. Certain types of quinones are there which can accept it here. And from here, it is being transferred to the FES complex. From here, it is transferred to ferredoxin. Okay, it is called as ferredoxin. And now, from ferredoxin, what happens? There is the formation of, you can see here, NADP plus H plus, it forms NADPH. Correct? You got it? So here, this is being formed by, there's a complex actually, it's called, abbreviated as FNR. This complex, correct? There is a reductase. This reductase actually helps in NADPH reductase. This reductase helps in the formation of NADPH molecule. Now, so you got this Z scheme with the electron acceptors. Okay, so please keep posting if you have any questions. We'll answer them. So far we have seen what are the components which are involved over here. Now we will talk about two important systems, that is cyclic photophosphorylation and non-cyclic photophosphorylation. Correct? If you can understand this sequence, अगर आपको ये समझ आ गया है, then we can talk about the cyclic and non-cyclic. First, let us talk about non-cyclic because you have already discussed about it. I'll show you how. So non-cyclic photophosphorylation. So this is basically called photophosphorylation because with the help of photon, phosphorylation process is happening. Okay, that means this ATP molecule is formed from ADP, phosphate is added, so it is called phosphorylation, right? So this photophosphorylation is happening in this Z scheme. If it is happening in Z scheme, you see it is non-cyclic. Why? Because the electron from here, it is being transferred at this phaophytin. Next, slowly and steadily, it is being transferred to photosystem 1. Then again, it ended with the production of NADPH and ATP. Got it? So if I ask you that what is the product for the non-cyclic type of photophosphorylation, it will be one ATP molecule and one ATPH molecule. Correct? Now we'll talk about the next type, that is cyclic photophosphorylation. Now let's see how this happens. Now you have to understand where this happens first of all. So in the chlorophyll, we have seen the grana molecules. Okay, the thylakoids are there, and each of these grana, they're connected by the stroma lamellae. Correct? If you remember, stroma lamellae helps them in the connection. I can show you. So here, these are the grana connected by the stroma lamellae. This is the stroma lamellae. Okay. Now this grana, it has the membrane of this thylakoids present in the grana. It has both photosystem 1 and photosystem 2, as well as they have this enzyme which is NADP reductase. Correct? This reductase is necessary so that this NADPH is formed. But unfortunately, in this stroma lamellae, only photosystem 1 is present. So what is present over here? Photosystem 1. So photosystem 1 is just present over here. So you understand, if only photosystem 1 is present, that means we can say that only this part is present. If this part is present, that means from where will it get electrons? Correct? So it cannot form a process on its own. Now what happens when sunlight falls in this photosystem 1 in stroma lamellae? Electrons which were initially there, already present, they are being excited. It is being passed over here, and what happens, it follows the same sequence से यहाँ पे पहुँचता है पर, like from this ferredoxin it transfers to what? I'll use a different color, maybe we can use this brown one. It transfers to plastoquinone, and in this plastoquinone ATP is produced and not NADPH, correct? ADP plus PI. So it produces ATP, and this part of the reaction is not happening. You understand? And after this, the electron comes back to the photosystem 1. So what did it become? If I close these parts, so does it seem a cyclic process? Correct. It is a cyclic process. So can we discuss once more? So non-cyclic photophosphorylation was that which we can see it over here. Here from photosystem 2, electrons are being excited. It comes back to photosystem 1, again excited, it comes back, and at the end, the result is that NADPH is produced. Now NADPH is produced over here. When in these stroma lamellae, there is only photosystem 1 and there is no NADPH reductase. If there is no NADP reductase, then what will happen? It cannot form this NADPH. So what happens? Till ferredoxin, the electron is transferred in this process, and then it is transferred to plastoquinone, and in this process ATP is produced. So the ultimate result of cyclic photophosphorylation, if it is being asked, you have to say it is one ATP molecule is produced, correct? So this is a cyclic process. Now, how energy is being produced? Let us quickly talk about how energy is being produced with this NADP and NADPH. So you have to remember that NADPH and ATP is produced over here. So let's continue our discussion with how the next process is happening, that is the formation of, there is a process which is called as chemi-osmotic hypothesis. So here we will try to understand how actually ATP is produced. So we are talking about ATP. So this ATP which is being produced over here, let's see how actually it is produced. So we'll do it diagrammatically. So here I am drawing, this is the membrane bound thylakoid. See, double membrane thylakoid. Now in this membrane there are three systems. Okay, so what happens? There are three systems over here. There is photosystem 1, photosystem 2 is first, then there is a complex called, we have seen, cytochrome B and F, correct? You get it? And there is the photosystem 1. Now it has both the systems because we are talking about grana. Okay, now we were talking about photolysis, that means production of H plus and electrons. Now this process of photolysis happens in the lumen. So this is the lumen, right? And this is the outside part, that is the, we can say, stroma. So in the lumen, what is forming? We are seeing the formation of H plus ions. Now why? These H plus ions are found because of the process of photolysis. So remember, because of this photolysis, H plus ion concentration is increasing over here. Now, when the photosystem starts, the electrons are being transferred through this photosystem 2, then cytochrome complex, then photosystem 1. We have studied that. But these electrons are not transported to an electron acceptor, but actually it is transported to a hydrogen carrier. Correct? So I repeat, these electrons are not transported to an electron acceptor, but they are actually hydrogen carrier. That means that when from photosystem 1 it is being transferred, the electron is being transferred to this complex, what happens? The H plus ions which are present in the stroma. So you understand, H plus ions are also present in the stroma, and stroma is the liquid part of the chloroplast, okay, the cytoplasmic part, you can say, of the chloroplast. So in this process, what it will do, it will use H plus and the electron is accepted. In this process, it will pump an H plus inside. So H plus is pumped inside, correct? I repeat. So in this process, electrons are not transferred to an electron acceptor, but it is transferred to a hydrogen carrier. So in this hydrogen carrier, this process, H plus ion is inserted here. And also, the enzyme for the NADPH reductase is present in this membrane, outer membrane, that is towards the stroma, you can say. So here, NADPH is produced from DP plus H plus. Got it? So in this process, this NADP reductase is present outside towards the outer membrane. Again, in this process, H plus is being taken. So what happens? Inside the lumen, the concentration of H plus is, it is LUMEN, lumen. Concentration of H plus is increasing first because of the fact of photolysis of water. Secondly, because the complex is pumping, these are proton pumps. They pump electrons, they pump protons, correct, from the stroma. That means the proton concentration outside the stroma is reducing and inside it is increasing. And also, thirdly, protons are being used by this NADPH reductase. So that means that in the stroma, the concentration of H plus is decreasing and in the lumen, it is increasing. This is happening. So what will it create? It will create a proton gradient, or we can say it as the chemical gradient is created. Now because of the increase in H plus, you understand the pH will also reduce because it is becoming acidic because of the presence of H plus. Now happens the wonder. Here we have a complex which is the ATPase system. It is called ATPase system, which has got two components. Okay, this is the F0, this is called the F1. This F0, because of the presence of H plus, what it does is it actually acts as a transmembrane. It acts as a facilitated transporter. It transports this hydrogen ions through this, and when it transports, it produces enough of energy for this F1 to function, in which what is formed? ATP, ADP to ATP, correct? So you understand, this proton gradient is required so that this ATPase starts functioning, in which the F0 will try to pump H plus, and because of this pumping, enough of energy is generated in which ATPase system will produce ATP. So let's see. Okay, so I hope you have understood this part. Okay, this is called the chemiosmotic hypothesis, in which you see this chemical gradient is very much necessary so that this ATP is synthesized, correct? So here in today's session, what we have talked about? We have talked about two important photosystems. We have talked about the light harvesting complex. We have seen the photosystem, the working of photosystem 1 and photosystem 2. We have also talked about the electron transport that is happening in these photosystems. Also, during this electron transport, we have talked about that ATP is produced, and now we have also talked about this chemiosmotic hypothesis, in which we have tried to explain how the ATP is produced. Okay, so because of this ATP, we'll see that how this ATP and where this ATP and NADPase is used further in the process. Actually, it is used to do other processes, which is basically happening in the stroma, in which the starch molecule, okay, the carbohydrate, the starch molecules are prepared with the help of this energy. So if you have questions, please quickly keep on posting so that I can solve your doubts, right? So before that, maybe we can, till you pose the questions, I can put up some certain quick questions. Let's see if you can answer them. So what are the two systems we have talked about? Quickly, two systems. So we have talked about two systems, that is photosystem 1 and photosystem 2. Now photosystem 1, what is the wavelength of light used by photosystem 1 for its excitation? Quickly, it is 700 nanometers, correct? Okay, What about photosystem 2? What wavelength of light is being used by photosystem 2? 680, correct? Now if you talk about the arrangement, I can show you the arrangement once more. So the arrangement we have talked about, this is the arrangement we have seen. So these are the electron acceptors. First it starts with photosystem 2 and then it goes to photosystem 1, correct? What are the two types of photosystems or reactions? Photophosphorylation we have discussed. One is cyclic, second one is non-cyclic, right? Non-cyclic is the Z scheme we have talked about, and cyclic is the one which happens in the stromal lamellae, and it happens just for which photosystem? It happens for photosystem 1, correct? And it happens in this way, since it is called, and hence actually it is called cyclic photophosphorylation, correct? So again, quickly before we end the session, if you have any questions, please post it so that I can solve your doubts. Okay, so I hope you have understood this session. So again, come back to Toppr.